Right, so good afternoon, everybody, and thank you for joining us. My name's Rosemary Collier, and I'm the person who's been emailing you. So this is about I think the sixth webinar in our monthly series. I think we'll be taking August off, and this time Dr. Stephen Parnell, who's a relatively new colleague in Warwick Crop Centre, is going to talk about outsmarting the outbreaks, predictive models to protect plant health. So just to point out that we'll be recording the session, and the recording will be available later. We've been awarded 1 basis point and 1 neurose point, so I'll send an email after this, and then you can respond to that if you want the points. And we'll take questions at the end of the presentation, but feel free to type questions into the Q&A or to the chat as we go along. Right, so I'll unshare and hand over to you, Stephen.
Okay, thank you. Okay, so you should be able to see my screen now, Rosemary?
Can do, yeah, brilliant.
Okay, so good afternoon, everyone. Thank you for joining, or thank you for watching if you're watching the recording. Thank you also to Rosemary for the opportunity to talk at this webinar in the series at the Crop Centre. So my name's Stephen Parnell. I'm a plant disease epidemiologist and a modeller here at the Warwick Crop Centre. And I'm going to be talking about some work that we've been doing on the use of predictive models to detect and control outbreaks. So the talk is called Outsmarting the Outbreaks, but I'll leave it up to you to decide who's being outsmarted by the end of it. So this is the problem. This is a graph that many would have seen from Ian Boyd, who was DEFRA chief scientist before Gideon Henderson. And this is showing the rise in the number of introductions of plant pests and diseases over the last few hundred years. So by both microbes, pathogens, and also insect pests, shown for the US in green, the UK in blue, and Europe in red. And what you can see is this sort of increasing trend over the last sort of 100 years or so. And this has been attributed to many different factors: climate change, the way we're moving plants around the planet, and accidentally moving pests and diseases with them. And if we move into have a look at the UK, so this is an example for forest health in the UK. So this is from the UK plant biosecurity strategy. And you can see, again, the cumulative number of introductions of different tree pests and diseases over time, this time since 1970s. And I've added just some examples on the y-axis there of some of these introductions. So people will remember Dutch elm disease from the 70s, but then you can see after 2000 this really rapid escalation in the number of introduced pests and diseases that the UK trees are facing. This is a global problem. So this is a paper that we're about to publish in Phytopathology. So it's currently, I think, in the early look. But this was work done with Cabby and with Mike Yeager, where we looked at both a systematic review of the literature to find surveys of plant pests and diseases, and also interrogated the Cabby database on records of plant pests and diseases. And essentially what you can see is that this is a global problem. This is just diseases, so this is not insect pests, but they range from fungi, viruses, nematodes, bacteria, and across a wide range of different host plants. So the cereals, forestry species, fruits and berries, vegetables, we have everything there. So what is at risk? Well, simply put, the environment. for one, on all of the different cascading impacts that come from that, the different ecosystem services that different natural plant communities support. And of course, also food, where pests and diseases are impacting on agriculture. So that might be impacting on agricultural economies, the price of food, but also food security at a time where global hunger is actually on the rise. So the work that we're doing at Warwick Crop Centre, the overall aim is to use predictive models to tell us where and when these outbreaks are most likely to occur. And the idea then is that we can use that to then target our surveillance, our eradication, or our disease management or control strategies. This plot here, I'll explain a little bit in more detail later in the talk, but it just gives you an idea of what we're trying to do. We have two things here. We have a disease spread model, and then on top of that, we're running a machine learning algorithm that uses the outputs from the model to look at where should we put locations in the landscape for search if we're trying to detect an invading pest or disease. And I'll come back to that graph and explain it in more detail later in the talk. These are some of the examples of outbreaks that we're currently working on. And in the webinar, I'll be talking about just a few examples here. But we've got everything from tree pests and diseases that you can see along the top here. So ash dieback is an important disease that's in the UK and much of Europe at the moment. And we're currently worried about the introduction of emerald ash borer, which is currently in Russia and Ukraine, but is a good hitchhiker and could be introduced to ash, which is already quite vulnerable because of ash dieback. Right the way through, so from tree health problems, outbreaks, right the way through to work that we're doing on cassava. So this is a staple food crop in Africa. 500 million people rely on this as a staple food in Africa. This is we're working on a project in West Africa, which is threatened in two ways. One by a virus, cassava brown streak virus, which is coming from East Africa and spreading into West currently in DRC. And then secondly, from a particularly severe strain of cassava mosaic virus, the Ugandan variant, which caused up to 4,000 deaths and recurring famine in East Africa in the 80s. And that unfortunately has just been found in Sierra Leone. But the first example I'm going to start with is spotted lanternfly, which is a project that we've been doing with the California Department of Food and Agriculture. So you can see from the pictures here, spotted lanternfly. It's found on a wide range of different crops. It's quite polyphagous, so it's on crops, it's on tree species, plants, particularly found in urban environments. And I give just a few examples there. So apple, prunus, grapevine, birch, Vagus pinus, oak. So a really wide variety. I think it has around 150 or 200 different known host species. And on some of these host species, it can be very impactful. So for example, on grapevine, it causes up to 90 to 95% yield loss. Its preferred host is the tree of heaven, and where it completes its life cycle. And this is actually an invasive plant and is distributed, and you can find it all over England and Wales, but particularly in the south. And the pest can cause so much damage because they kind of come en masse onto their hosts and can be quite destructive. So it's native to China, but it was found in the US for the first time, I think in Pennsylvania, in the east of the US, and it has spread gradually ever since. But, and this is a study by Wakey et al. 2020, looking at the potential distribution. So they're using a model called Maxent to look at where in the world could it survive if it was introduced. So obviously we know it's in China, we know it's in the east of the US, but then using data, looking at the climatic variables in those areas, where else could it survive? And you can see that there's quite wide potential establishment across Europe, including some low and medium establishment areas in the UK, but also in California. It's been earmarked as quite high potential establishment because of the climatic conditions there. So the project with California Department of Food and Agriculture, they were interested in two questions primarily. First of all, trying to understand what is at risk in California. So there had been studies done looking at the different crop species and what their value is in California, but they hadn't taken a biological approach looking at, well, it might not be able to establish in all of the areas where all of those crops are grown. So we wanted to look into that in a bit more detail, because although California as a whole is suitable for spotted lanternfly, there is some variability associated with that. So you can see from this map here from Jones et al. 2022, there's patchy establishment risk across California, some potentially quite high risk in some areas, lower in others. The second question that CDFA were interested in is, well, as far as they know so far, spotted lanternfly has not been found in California. It's not in California. So where should they conduct early warning surveillance so that if it does arrive, they can detect it as soon as possible and hopefully have successful eradication measures implemented? So we started with, this is quite a straightforward approach. So we just looked at the host crops in California. There's 22 cultivated crops that are host species. And we looked at their area and their average value across California. And then we took this with models of establishment risk, looking at if the spotted lanternfly landed in any particular area in California, what would be its reproductive number, how much could it actually expand, and the variability across that. And we combined that with data on the severity. So it is particularly bad on grapevine, but it's got then variable severity on other host species, and we looked at some scenarios for that because there's some uncertainty. It's certain that it's bad for grapevine, but there's some uncertainty about its impact on other host species. And what you can see, so here I've zoomed into two counties in California, San Joaquin, which is in further north, and Kings County, which is a little bit south. And you can see that there's both variability between counties in California. So either very high climatic risk, or it can, in some counties, much lower. The cultivated host map on the left, so the yellow there is grapevine. The red is walnut, I think, and the green that you're seeing there is cotton, which is well, quite a minor crop in California, but still grown in some areas. And we modeled this and we took into account all of these different factors. So the host area, its value, the severity of the pest should it arrive on that particular host, as well as modeling of the climatic risk, together with Temple University. Matt Helmus's group there did a lot of that work on the climatic risk. And if you combine this information, you can come up with more accurate estimates of what the value at risk is. So although some areas have crops like grapevine that are particularly at risk, not all of that is in areas that is suitable, for example, for the pest. So you see a lot of variability between counties, but also within counties as well. There can be a lot of variability because of the topology and the climatic conditions within the different counties. Then we took this one step further and we looked at, well, how can we use this information that we've got on how quickly it might bulk up, depending on which area it arrives in. And then we also included some information on entry risk. So here we used information on human population density because the egg masses can be of spotted lanternfly can be moved around, as well as infested plant material. So really a good proxy for entry risk is where you have overlapping areas of high human population density, but also that is adjacent or near to areas of where there's host plants for the crop. So if we look at where those things overlap, both high population density but near to host plants, we're able to come up with an entry risk map. Then we combine that with the other information that we have on the value at risk to come up with a surveillance suitability metric. And then the dots on here are CDFA would say, well, we want to look at 100 different sites. So we can then do a weighted random sample of that surveillance suitability metric to look at where they would be best to concentrate those surveillance measures to have the best chance of detecting an invasion at an early stage, because surveillance resources are obviously quite constrained, and California is a large place. So being able to use these sorts of approaches to really target surveillance is important. Okay, so the next example I want to give is work that we've done on citrus greening. So this is work that a long-term collaboration with the US Animal and Plant Health Inspection Service and the USDA in Florida and in California, and also work with EU collaborators within Europe. So Huanglongbing, or citrus greening, so Huanglongbing means yellow dragon disease in Chinese because the symptoms in the foliage create this, apparently look like a sort of dragon spreading through the citrus trees. It's a bacterium that's spread by this little guy, this Asian citrus psyllid, and there are some other vectors that are in Europe. So 2 vectors are in Europe at the moment. But in North America, it's primarily spread by the Asian citrus psyllid. And it's a pretty devastating disease. So the first HLB positive trees were found in Florida in 2005. And I worked for the USDA in Florida, and I also arrived in 2005, but a complete coincidence. But HLB was first found in 2005, and then you can see, so in Florida, the commercial citrus is really concentrated in the middle of the state. And you can see what happened. So this is a map of positive locations from 2006 up to 2012. And really what this is showing is that almost every citrus tree was infected over a 10-year period. So it's had a devastating impact on the industry. So the Florida citrus industry is now down to about 15 or 20% of what it was in 2005. So not 15% less, but it's at 15%. So the industry is almost vanished or vanishing from Florida. So there's a huge concern about this in other citrus producing areas of the world. So of the 10 major citrus producing areas of the world, HLB is already in 7 of them. It's not in Europe. So there's work being done in preparedness planning and surveillance in Europe. It is present in California, but at the moment it's only in residential areas. So citrus trees that people have in their back gardens. And because they've had huge monitoring programs in place and huge programs of spraying to control the vector populations, so far they've been successful in keeping it in those residential areas. So we had some data from Florida, so we were looking at this retrospectively, really, because it's already obviously invaded in Florida and spread everywhere. But we looked at this question of, well, can we utilize the data to learn what could have been done or what should have been done in Florida to try to detect it, to put surveillance in place so that when it did arrive, it could have been detected at as an early as possible stage. So we had as inputs to the models, we had data on the citrus density. So as I said before, so commercial citrus is in sort of the center of the state, but many people have citrus in their backyards in Florida. So there's a high citrus density in residential areas as well. But we had that data. So the commercial citrus inventory is known. The residential, they did a lot of work where they went into different residential areas of different types and counted the citrus trees, and then were able to then estimate distribution throughout other areas based on the different residential types. So the first thing was we knew where the citrus was. The second, we had a map or a model for the risk of introduction of HLB. So the USDA developed something called the travel census model, where they essentially the way the model works is they look at areas of the world where the disease is endemic, and then using travel like the customs forms and census data, they know where people are in Florida that have a high level of connectedness to those disease endemic areas of the world. So we combined these bits of information and we developed a spread model, and you can see it just running there on your screens. So what it does is it's stochastic, so it runs a different realization every time. We'll run 1,000 or 10,000 of these to get an overview of all the possible behaviors of the model. And every time we run it, we start it from a different location, but weighted towards this risk of introduction map. And then this is the figure that I just showed at the beginning of the webinar. You can see what we did was from the spread model, from the predictive model, if we're going to look at, say, 50 locations in the landscape, for any one set of 50 locations, from the model, we can calculate what the probability would be to detect the disease. So what the optimization algorithm, so this is a machine learning algorithm called simulated annealing, and what it's doing is it's moving, it starts with a random set, it moves one and it sees did it improve the probability of detection. And then it will accept all positive changes, it will accept some negative changes just because otherwise it can get stuck in local minimum. But eventually it finds the set of locations that maximize our probability to detect the epidemic. So what you can see on the right-hand side, that the optimal strategy far outperforms the random strategy. So that would just be randomly allocating resource, but it also outperforms some other strategies. So one thing, and this is often what happens, is people focus around entry risk areas. And that does okay, but even better would be to focus on the areas of highest citrus density, at least in this example, because that's where it will be able to spread and bulk up quite quickly. But the best kind of rule of thumb approach is to use a combination of those two things. So where you have overlapping risk of entry and high citrus density, that's what will get you closest to the optimal solution. A few other insights from this approach is that it showed that the optimal solution was often to distribute your resources actually quite widespread in the landscape. So you can't really see it here that well, but there's really in Florida, there were two main risk areas. So one in the Treasure Coast and then one just north of the Everglades. So if you just ranked from high risk to low risk, the highest risk areas will be all in just those two clusters. But what the algorithm is saying, what the model is telling us, is that we need to spread our resources a bit more widely than that. And this kind of like table here sort of shows why. So it depends on our objective. So if our objective was just to try to maximize the number of distinct finds, so just for all of our cases, just find as many positive cases as possible, then what you could see here is that we should focus on site 1 and site 3. So this is just a toy example where I've just got 6, imagine a landscape of just 6 different sites that we can look at or that the disease can get to, and we've run the model 8 times. The X marks where in run 1, site 1 was infected but no other sites. In run number 6, site 4 was infected and site 6 was infected. So this really captures the uncertainty in the model, because in any one run, any one sort of like realization, we don't know exactly what's going to happen. But if we wanted to just maximize the number of distinct finds, then the sites that we should look at are site 1 and site 3. However, if what we want to do is just find it once as early as possible and raise the flag that it's here, so just find it at as early as possible prevalence, so just that we know it's now in Florida and then we know that as early as possible, then actually the optimal thing to do is to look at site 1 and site 4. And you can see it's because of this correlation. So if you look at site 1 and site 4, you catch it in every single run of the model. So you don't need to look, although in sites, say 1 got infected the most 6 out of 8 times, you don't need to put more resources into that than necessary. You only need to look there once in order to know that it's there, so you can afford to spread your other resources more widely. So that was the first insight from the model. The second was that how good you are at detecting changes where you should look. So how good you are at looking changes where you should be looking. So if you have when you do look, you look very, very intensively, and you have a detection method that has a very high sensitivity, you can afford to look more broadly. But if your detection methods are not as effective, or the pest is more cryptic and difficult to detect, then actually it is better to just focus in on these 2 higher risk areas. So that's early detection, sort of finding it in an area for the first time and sort of raising a flag that it's here. I'll just talk briefly now about some other work still on citrus greening, and this is work with EFSA. When we find a pest for the first time, the next question that is asked is, well, how far has it spread? Because usually what you will do is set up a containment area where you're going to apply phytosanitary measures. And what we found when we looked at this with EFSA was that actually the way people delimit that area, that infested area, is quite ad hoc, and everyone does something different. So we were interested in, well, what's the reasons behind that? How do different approaches that you can take to delimitation vary? And we compared 3 different approaches. One was an in to out method. So that just means that we take the first site that we found that's infected and we just start moving outwards in bands until we can't find it anymore. The second approach was what we called the EFSA approach, because what we ultimately ended up using was where you look at how long do you think the pest has been spreading for. So that might be determined by, for example, the time since the last survey. So let's say you've had 5 years since you conducted a survey in this area. You would assume that you've had 5 years worth of spread. You go out that far, and if you still can't find it, you keep going out sorry, if you don't find it, you start moving inwards. But if you do find it, then you start moving outwards. So it's a sort of adaptive strategy that takes into account some of the information on spread. And then the third one was what we call the multi-foci strategy, which is where every positive find you draw a new circle, and you end up with this area of overlapping circles. So we tested these different strategies with the model, and in particular, we're focusing on Europe, and we were looking, what I'm going to show anyway, is a focus on residential trees. So residential trees in Europe are mainly in streets and parks, whereas in Florida, they're in backyards. So we looked at different types of landscape topologies, and we modeled the spread on this and tested out the different strategies. And what we found is that the in-to-out strategy and the EFSA strategy of moving outwards and then being adaptive. So don't worry about too much of the detail on this graph, but really the red and the blue, which represent these strategies of having just one circle, outperform the multi-foci strategy in terms of both their effectiveness in terms of how much of the area, the infected area, they capture, but also in what I'm not showing here is also they're more resource friendly as well. Okay, so the next example I'm going to show is Xylella. Actually, just before I do that, I'll say that what you can also take from this is that there was no one strategy that's perfect. So there's a lot of variability depending on where the epidemic happens to start and how fast it spreads. But you can see there's not a perfect strategy. And part of the problem for that was that we were assuming, which is often the case in practice, that surveillance was based on visual inspection. And for this disease, it has a long asymptomatic period, and that made accurate delimitation very, very challenging. And the reason I sort of emphasize that is because that's sort of also an issue with this next outbreak that I want to discuss, which is Xylella fastidiosa. So this is a bacterial disease spread by these xylem sap-feeding insects. It was historically confined to the Americas, but it was found in Europe for the first time in 2013 in the heel of Italy in Apulia. It's an has an extremely wide host range. Sorry, I'll just go back. And it's very impactful. So there were demarcated areas set up in Apulia and emergency legislation in place to try to eradicate the pathogen. And it's since been found in other areas of Europe on other host species as well. So one of the things we looked at for this was this problem of visual inspection that I mentioned for Huanglongbing or citrus greening. So what this graph here shows is the prevalence of visual signs of disease on the x-axis. So what we did was we ran a model and we calculated what would be the prevalence of the disease when it's first found. And then we back calculated what's the true prevalence of infection. So for something like Phytophthora ramorum, you can see that if you detect it when there's 5% of symptoms showing, there's about 5% of level of true infection. So it's not so different. But for Xylella on olive, when 5% of trees are showing symptoms, the true infection is 75%, so much, much, much higher. So really visual inspection in that case, at least in terms of early detection or eradication, is not very helpful. And you can see Huanglongbing that I've just been talking about, it's not quite as bad, but still quite severe. So we were interested then, well, if you can't use visual inspection, what methods can you use? And so we looked at comparing sampling hosts versus sampling vector populations. So capturing the vectors and testing them for presence of the bacteria. So we used data on monitoring from Italy on vector infectivity and also host infectivity. And we used that to parameterize a model. And essentially what we found was that it would be, for the examples that we were looking at, it would be more cost effective for early detection to sample vectors than it would be to sample hosts, because you can pick it up earlier in the transmission cycle. We've also done some work, so there's been some very successful work by people like Peter Beck and Carlos Camino at the JRC using remote sensing to pick up Xylella, and again tackling this issue of this long asymptomatic period before symptoms are expressed. And they found that by using hyperspectral cameras and thermal imagery, they could detect it from the air before you could actually see symptoms on the ground. So that was quite exciting, but what we did was then we tried to look at, well, the remote sensing treats every single pixel as if it is independent and it's completely ignorant to what's going on in other pixels. So we combined the remote sensing estimates with a predictive spread model that basically says that hosts, for example, like the ones you can see near the bottom right of the map here that are close together, they're more likely to have a shared disease status. So the spread model captures how the disease can move through the host trees in the landscape. And we found, so this is just a measure of the accuracy before and after the spread model. And essentially, if you focus, for example, on the right-hand graph, so don't worry about the details too much, but essentially what we found is if we coupled the remote sensing with the spread model, we could achieve higher levels of predictive accuracy just by including that bit of biology that tells us about spread. So we've also looked at the spatial optimization for Xylella. So we have this demarcated area here in Apulia. The question is, well, where should you conduct surveillance outside of this infected zone, because you want to know if it's escaped or not. And we considered two different scenarios. So the bottom row essentially shows a scenario where we don't include human-assisted or long-range jumps. And what you can see there on the bottom row is that if you do that, then the optimal thing to do is just keep most of your samples in this outlined area, which is the buffer zone. However, on the top row, what we found is that even if we included a small possibility that Xylella could not just spread naturally, but jump occasionally longer distances, then the strategy completely changed. And you can see that in the top right. Then the optimal thing was to look much more broadly and not just in the buffer zone. Because although natural spread leads to sort of bulking up and sort of this gradual movement of the infection front, these rare long distance jumps cause new foci and new outbreaks. So even if they're rare, they can be quite devastating. So surveillance in more widespread than just in confined areas and buffer zones is quite important. And the final example I want to show is work on acute oak decline. And this is the motivation for this is getting at this problem of detecting these outbreaks at an early stage is a bit of a needle in a haystack problem. So bringing in new technologies like remote sensing, but also different surveillance sources like volunteered data can really increase the numbers on the ground and lead to a better chance of detecting these things at an early stage. The UK is, I think it's correct to say, a world leader in citizen science in plant health. So I was involved in an EFSA project where one of the tasks we had was to come up with an inventory of citizen science programs across Europe. And there are by far and away the most citizen science programs in plant health are in the UK. And that's thanks to these sort of programs like Observatory, funded by people like DEFRA, Woodland Trust, APHA, that really engage volunteers, train them, and also mechanisms like the use of smartphones, websites like the Forestry Commission's Tree Alert that give people a mechanism to report what they're seeing. So citizen science, or this volunteer data, is not just one thing. So this is a nice figure from Susan Hester, looking at invasive species citizen science efforts more generally. But she conceptualized it as a kind of continuum from passive to active. So passive on one end of the extreme, where people might just be out and about in woodlands and they might spot something and then report it, right the way through to active surveillance, which is often conducted by MPPOs or specific agencies that will be searching for plant diseases. But you have other things in between. So you have citizen science groups that are maybe doing something more structured than just reporting things ad hoc. As well, you have landowners and farmers that have opportunities to spot things. So there's lots of information sources beyond official surveillance that could really make a step change in our ability to detect things at an early stage. But there's challenges associated with it. So for one, it's if you want to interpret this data, you need to have some sort of idea of the surveillance effort that's going on. So, for example, if a member of the public reports something, a new pest that isn't known to be in the UK for the first time, what does that mean? If there's very, very little awareness about that pest or disease, then the chances are it could have reached quite a high prevalence before just one person or a small number of people would actually report it. But if there's a lot of effort, people are aware of it, a lot of people looking, it could be at a lower prevalence. So we need to know that. And then we also need to know, well, if someone is confronted with the pest, what's the chances that they will correctly identify as the pest? And that's sensitivity. And equally, we need to know, well, if someone makes a report, but it's a false report, then we need to know what the chances of that are, and that's specificity. So you'll remember these terms from the good old COVID lateral flow tests, where their sensitivity and specificity were constantly under scrutiny. So we focused a lot of this work on acute oak decline. It's an important pathogen or syndrome, a condition of oak trees in the UK. And what we found in some initial work was that there was a structured survey done of England and Wales of acute oak decline, and there was also ad hoc land reports from landowners. And what we found, interestingly, was that they both, when you looked at them, they both identified a similar area as being infected. And we're able to come up with an approach to combine it. But I think it gave us a lot of faith in this sort of data. And then more recently, what we've been doing, so in summer in 2022, Nathan Brown and Peter Crow at Forest Research conducted some experiments with volunteers in acute oak decline woodlands. So Nathan runs some monitoring sites where they have been monitoring acute oak decline for, I think, maybe 15 or 20 years now. And they have an idea of, for every single tree in those study sites, the symptoms that are there. So what the volunteers did is they went into these two different sites. They each assessed around 100 trees. So we had nearly 2,500 individual tree observations, and they scored them for the different symptoms of acute oak decline. So we had two data sets. We had one, our expert data set, or our gold standard of what the true disease status was, and we had what the volunteers thought. And they scored them for these three different symptoms, so stem bleeds, exit holes, and bark cracks. So the exit holes, because the bacterial infection is associated with an agaricus beetle that leaves this distinct D-shaped exit hole when it in the bark. So we were able to quantify the sensitivity and specificity of the volunteers. What you can find, if we look at exit holes, for example, you can see there's quite a wide range in sensitivity, partly reflecting that we had a wide range of volunteer types. So I think we had everything from teenagers that had been dragged along by their parents to kind of retired entomologists. So there was quite a lot of variability there. But you can see the specificity is actually pretty good. So although people might miss an exit hole, they wouldn't say there was an exit hole where there wasn't. And I guess it's because they're difficult to mistake with other things. But the important thing is that we know what these values are, because we need to know them in order to interpret the data and to be able to answer questions like, what's the likely prevalence of a disease? But the problem, of course, is that, well, in this case, we had our expert data, so we could calculate sensitivity and specificity because we had our validation or our gold standard data set. In many cases, that data, unfortunately, won't be available, but we'll want to know what the sensitivity and specificity of volunteers or other observers is. So Matt, who's a postdoc in the Crop Centre, looked at this, and he developed an approach that was based on a Bayesian modeling method that had originally been applied in veterinary medicine. And he was able to develop this statistical approach, which essentially compares volunteer observations with each other in order to arrive at an estimate of sensitivity and specificity. So we can estimate these values without actually knowing what the true values are. And then in this AOD case, we could then see, well, how well was this doing? Because we had the actual values, and it's doing, you can see it's doing pretty well. So there's a lot of hope here for like increasing numbers on the ground and actually being able to do so in a way that we can use this data to better detect some of these different outbreaks. Okay, so I'll stop there, but just to say that hopefully I've got across the sort of importance of sort of science-based and model-based approaches to surveillance. Because they allow us to take a kind of risk-based approach that can be more targeted, more chance of success, and sort of optimized. And there's ways of including new technologies, new sources of surveillance information into the models to then improve these further. And I'll just start by thanking my collaborators on this work, in particular Matt and Joshua, PDRAs, that have done a lot of this work at Warwick, and also the funders. So thank you for your attention, and hopefully there's some time for questions.
Brilliant. Thank you very much, Stephen. That's great. So has anybody got any questions? I don't think there are any typed in at the moment. If you'd like to ask Stephen a question, then yeah, put your hand up, and I think we can unmute you in this system. Oops, there we are. There's one here. I'll just unmute you. Edward Birchall should work. Can you speak now, Edward? No.
Yeah, hopefully.
Oh, brilliant. Yeah, you can.
Yep, I can hear it.
Well done. You've pressed the right buttons.
Yeah. Right.
Excellent.
Fire away.
Thank you. Lovely, good, excellent talk, Stephen. Very interesting. I work for APHA and have done in eradication, containment, and other parts of my roles have changed over the years. But when you were talking about the HLB, of particular interest is the models. Do they take into account things like the variability of weather patterns, as we know that certain organisms will blow particularly very well, and we've had that occurrence happen in GB from the continent?
Yeah, so it depends on the pest or the disease. I think in Florida we didn't, because the conditions are climatic conditions are pretty homogeneous across the entire state. It's not like that in Europe, so the models that we're applying there are taking into account, because obviously there's areas of, say, Spain that are mountainous and areas that are much more suitable. So in Spain, yeah, that becomes more important. So there's different ways that we approach it. So the simplest is just to include suitable and unsuitable areas using climatic data, or if needed, you can do something more using the climatic variables to sort of drive what the actual prevalence might be. So that's sort of ways which we can include it. The other thing I would say is that for diseases that are fungal diseases in particular, there's been work with collaborators looking at, for example, using the Met Office name model to look at not sort of how do the environmental conditions affect spread, but more like long range dispersal. So for example, on wheat rust, I think they've been using those sorts of approaches. Does that kind of answer the question?
Yes, yeah. Thank you very much.
Thank you. Any more questions? No? I was just going to ask you, Stephen, might be a silly question. Oh, Kim's going to ask a question, but I'll just ask you mine quickly. And then, Kim, I mean, what you've been talking about is surveillance, I guess, really. And I wondered whether sort of models were useful for management.
Yeah, so I've kind of we do a lot of that. I've kind of chosen to focus on surveillance here, because that's a lot of what my focus has been. But yeah, absolutely. Just like you can use the models as a kind of testing sort of simulation engine to test out different surveillance strategies, they can be used to test out different management strategies. So for example, for Xylella, we were looking at so the eradication program involves, for every tree that's detected or plant that's detected, removing everything within a certain radius. And there's a lot of debate about, well, what length should that radius be? So people generally want it to be as short as possible, but if it's too short, you'll always sort of stay one step behind this kind of asymptomatic spread. So yeah, there's lots of other examples where, yeah, these sorts of models can be used to test out then, yeah, what the management measures would be as well.
Thank you. Kim, I think I've unmuted you with any luck.
Yeah, can you hear me?
Yeah.
Yes.
Yeah, great. Well, this is a question for Rosemary and Stephen, actually. So, Rosemary, better wake up. So I wondered whether you've got any plans together or ongoing work where you've got examples of what you're doing, Stephen, that in collaboration with Rosemary more around less to do with forestry, but more to do with UK or European agriculture, horticulture. I've sort of got silver Y in mind, that type of thing, silver Y moth. And I also wondered whether within your models, you could also be looking at spread, but taking into account impact where, for example, we're losing fungicide or insecticide actives, so that there's a sort of change in susceptibility because we're not able to protect crops from certain pests or diseases, or host resistance as well.
Yeah, no. So I a lot of what I've focused on was kind of I guess geared towards quarantine, what you describe as quarantine plant pests and diseases. But yeah, we are doing work on sort of other pest and disease problems, obviously just as big. Rosemary and I, we've got a proposal in on IPM, so we are working together. I don't know, Rosemary, if you want to add anything.
Well, only that I guess Kim, you mentioned silver Y, and the same thing sort of applies to diamondback moth, and there's a group who's working on similar things to what Stephen's been describing, novel pests to the UK, which is led by the Met Office, and I've been trying to persuade them for a while to start addressing those species, because I think there's a huge weather component in those, in terms of, first of all, where they spend the winter, and then also which direction the wind's blowing to bring them in. So I think there's a lot of potential, but I think it needs a meteorologist as well or several meteorologists to help with it.
Thank you. Yeah, that's really helpful.
Okay. Any more?
There's also a question from Andy Evans in a Q&A chat.
Oh, right. Can you? Oh, in the Q&A. It's good, that voice in the background. Thank you. So Andy asks, is Maxent the main engine behind most of the models you discussed?
Well, so Maxent is certainly a popular one for the sort of climatic potential, the potential establishment climatically. That's sort of like one of the key models there. So it sort of like feeds that ingredient. But a lot for these sort of outbreaks, where they're driven by dispersal processes, what we're doing is really we have a statistical description of dispersal. So it goes into dispersal kernel, which essentially it just says as you move further away from an inoculum source, how does the probability that the pest will move decrease with distance? So we tend to use those sorts of approaches, but that can be then kind of embedded on top of a sort of these sort of like Maxent or climatic outputs from these sort of climatic models. But a lot of what I've been describing is really dispersal focused, which takes a slightly different approach. But can be linked with those sorts of outputs, and when we have done.
Great. Right. Any more questions before we wind up? Doesn't look like it. I think we've got everything covered. Right. So very many thanks, Stephen, for a really interesting presentation. As I said at the beginning, we've recorded it, so we'll put it online. And just to say thank you to Alex Kelly in the background as well, because Alex sets up all the meetings and then I try to learn how to operate them. So we'll stop there. Say thank you all for joining as well. I think we're going to take August off, and then I think we're going to try and spread our wings a bit in the new year and ask some of our some other organisations to contribute to webinars as well. So you'll be hearing from me again, and as I say, I'll send an email about the basis points probably tomorrow morning. So many thanks again, and have a good summer.
Okay, thank you.
Thanks, Stephen.
Thanks.